Let's continue from the previous session about the role of P53, guardian of the genome. The TP53 gene may have more to do with the development of human cancer than any other component of the genome. The gene gets its name from the product it encodes, P53, which is a polypeptide having a molecular mass of 53,000 Daltons. In 1990, TP53 was recognized as the tumor suppressor gene that, when absent, is responsible for a rare inherited disorder called Li-Fraumeni syndrome, which we have already learned. So, next topic. Why is the presence of P53 so important in preventing a cell from becoming malignant? For one thing, P53 seems to bind a very long list of different proteins as well as DNA and is involved in a diverse array of cellular activities. In its best studied role, P53 serves as a transcription factor that acts as a crucial player in a cell's response to stress. When a cell contains DNA damage, P53 responds by altering the expression of a large number of genes involved in cell cycle regulation, apoptosis, or senescence. The importance of the transcription regulating role of P53 is evident, which shows the location of the 6 mutations most commonly found to disable P53 in human cancers. All of them map in the region of the protein that interacts with DNA. One of the best studied gene activated by P53 encodes a protein called P21 that inhibits the cyclin-dependent kinase that normally drives a cell through the G1 checkpoint. As the level of P53 rises in the damaged G1 cell, expressions of the P21 gene is activated. So, cell cycle arrest is not the only way that P53 protects an organism from developing cancer. Alternatively, P53 can direct a genetically damaged cell along a pathway that leads to death by apoptosis, thereby riding the body of cells with a malignant potential. P53 is thought to direct a cell into apoptosis as the result of several events, including the activation of expression of the BAX gene, whose encoded protein initiates apoptosis. Not all actions of P53 are dependent on its activation of transcription. P53 is also capable of binding directly to several members of the BCL2 family proteins in a manner that stimulates apoptosis. For example, P53 can bind to BAX proteins at the outer mitochondrial membrane, directly triggering membrane permeabilization and release of apoptotic factor. If both alleles of TP53 should become inactivated, a cell that is carrying damaged DNA fails to be destroyed, even though it lacks the genetic integrity required for uncontrolled growth. Several studies have shown that established tumors in mice will undergo regression when the activity of their P53 genes is restored. This finding suggests that tumor development continues to depend on the absence of a functional P53 gene even after cells become genetically unstable. For these reasons, the development of therapies that restore P53 function to P53 deficient cells has become an active area of research. The most advanced therapy based on this strategy involves injection into the tumor of an adenovirus that carries a wild-type TP53 gene. This approach has been used widely in China, but a similar adenoviral vector, Advexin, has not been approved by the FDA at the time of this writing. The level of P53 in the healthy G1 cell is very low, which keeps its potentially lethal action under control. However, if a G1 cell sustains genetic damage, as occurs if the cell is subjected to ultraviolet light or chemical carcinogens, the concentrations of P53 rises rapidly. A similar response can be elicited simply by injecting a cell with DNA containing broken strands. The increase in P53 level is not due to increased expression of the gene, but to an increase in the stability of the protein. In unstressed cells, P53 has a half-life of a few minutes. P53 degradation is facilitated by a protein called MDMA, which binds to P53 and escorts it out of the nucleus and into the cytosol. Once in the cytosol, MDMA adds ubiquitin molecules to the p53 molecules, leading to its destruction by the proteasome. How does DNA damage lead to stabilization of p53? Persons suffering from ataxia telangiectasia lack a protein kinase called ATM and are unable to respond properly to DNA damaging radiation. ATM is normally activated following DNA damage, and p53 is one of the proteins ATM phosphorylates. The phosphorylated version of the p53 molecule is no longer able to interact with MDMA, which stabilizes existing p53 molecules in the nucleus and allows them to activate the expression of genes such as p21 and BAX. So, some tumor cells have been found that contain a wild type of TP53 genes but extra copies of MDMA. Such cells are thought to produce excessive amounts of MDMA, which prevents p53 from building to required levels to stop the cell cycle or induce apoptosis following DNA damage or other oncogenic stimuli. A major effort is underway to develop drugs that block the interaction between MDMA and p53 in an attempt to restore p53 activity in cancer cells that react this key tumor suppressor. The relationship between MDM2 and p53 has also been dominated using gene knockouts. Mice that lack a gene encoding MDMA die at an early stage of development, presumably because their cells undergo p53 dependent apoptosis. This interpretation is supported by the finding that mice lacking genes that encode both MDM2 and p53 double knockouts survive to adulthood but are highly prone to cancer. Because these embryos cannot produce p53, they don't require a protein such as MDMA, MDM2 that facilitate p53 destruction. This observation illustrates an important principle in cancer genetics. Even if a crucial gene such as RB or TP51 is not mutated or deleted, the function of that gene can be affected as a result of alterations in other genes whose products are part of the same pathway as the crucial gene. In this case, overexpression of MDM2 can have the same effect as the absence of p53. As long as the tumor suppressor pathway is blocked, the tumor suppressor gene itself need not to be mutated. Numerous studies indicate that both the p53 and PRB pathways have to be inactivated one way or another to allow the progression of most tumor cells. Because of its ability to trigger apoptosis, p53 plays a pivotal role in treatment of cancer by radiation and chemotherapy. It was assumed for many years that cancer cells are more susceptible than normal cells to drugs and radiation because cancer cells divide more rapidly. But some cancer cells divide more slowly than their normal counterparts, yet they are still more sensitive to drugs and radiation. An alternate theory suggests that normal cells are more resistant to drugs or radiation because once they sustain genetic damage, they either arrest their cell cycle until the damage is repaired or they undergo apoptosis. In contrast, cancer cells that have sustained DNA damage are more likely to become apoptotic as long as they possess a functioning TP53 gene. If cancer cells lose p53 function, they often cannot be directed into apoptosis and they become highly resistant to further treatment. And this may be the primary reason why tumors that typically lack a functional TP53 gene like colon cancer, prostate cancer and pancreatic cancer respond much more poorly to radiation and chemotherapy than tumors that possess a wild type copy of genes, testicular cancer and childhood acute lymphoblastic leukemias. Then the role of p53 in the promoting senescence. We have seen how p53 can direct a potential cancer cell into either growth arrest or apoptosis. Recent studies indicate that p53 also controls signaling pathways that lead to cellular senescence. Another signaling pathway that leads to cellular senescence, another mechanism that has evolved as a barrier that stops wayward cells from developing into malignant tumors. So unlike apoptotic cells, senescent cells can remain alive and metabolically active but are permanently arrested in a non-dividing state as exemplified by the senescent melanocytes found in moles. And in other cases, senescent cells may be ingested by phagocytic immune cells. Senescence can be triggered in an otherwise normal cell by the experimental activation of an oncogene such as RAS, which might occur with some frequency during the day-to-day activities of dividing cells in a normal tissue. Studies suggest that oncogene activation triggers a period of accelerated division, after which the senescence programs take effect and slams on the brakes. This is the apparent route that is taken during the formation of benign molds. One of the pathways leading to senescence involves expression of tumor suppressor gene called INK4A, which is often disabled in human cancer, and INK4A encodes two separate tumor suppressor proteins, like proteins that are translated in alternate reading frames of the mRNA. P16, which is an inhibitor of cycling dependent kinase required for progression through the cell cycle, and ARF, which stabilizes P53 by inhibiting MDM2. The precise role of P53 in directing cells towards the senescent state remains unclear, but inactivation of the TP53 gene within senescent cells can cause the cells to resume their progress toward full malignancy. Then whether P53 moves a cell towards cell cycle arrest, apoptosis, or senescence apparently depends on the type of post-translational modification to which it is subjected. As in the case of the core histones, modifications include phosphorylation, acetylation, methylation, and ubiquitination of affect more than 3 dozen residues within the P53 molecule. Moreover, the fact that the TP53 transcript can be alternatively spliced into numerous P53 isoforms, that these P53 proteins can interact with a host of different proteins, and that P53 has been found to influence several other major tumor-related pathways. DNA repair, glucose metabolism, and autophagy add additional layers of complexity to the DNA to the P53 story. Dissecting the roles of these various factors in the function of this multitasking protein will be a daunting challenge. Then other tumor suppressor genes. Although mutations in RB and TP53 are associated with a wide variety of human malignancies, mutations in a number of other tumor suppressor genes are detected in only a few types of cancer. Familial adenomatous polyposis coli, or FAP, is an inherited disorder in which individuals develop hundreds or even thousands of pre-malignant polyps or adenomas from the epithelial cells that line the colon wall. If not removed, cells within some of these polyps are very likely to progress to fully malignant stage. And the cells of patients with this condition were found to contain a detection of a small portion of chromosome 5, which are subsequently identified as the site of tumor suppressor gene called APC. And a person inheriting an APC deletion is in a similar position to one who inherits an RB detection. If the second allele of the gene is mutated in a given cell, the protective value of the gene function is lost. The loss of the second allele of APC causes the cell to lose growth control and proliferate to form a polyp rather than differentiating into normal epithelial cells of the intestinal wall. The conversion of cells in a polyp to cells of intestinal wall or to malignant state, characterized by the ability to metastasize and invade other tissues, is presumably gained by the accumulation of additional mutations, including those in TP53. And mutated APC genes are found not only in inherited forms of colon cancers, but also in the major types of sporadic colon tumors, suggested that the gene plays a major role in the development of this disease. In its best studied role, APC suppresses the WNT pathway, which activates the transcription of gene like MYC, NCC, ND1 that promote cell proliferation. APC may also play a role in the attachment of microtubules to the kinetochores of mitotic chromosomes. Loss of APC function could therefore lead directly to abnormal chromosome segregation and aneuploidy. The presence of mutated APC DNA has been found in the blood of persons with early stage colon cancer, which raises the possibility of a diagnostic test for the disease. It is estimated that breast cancer strikes approximately 1 in 8 women living in the United States, Canada, and Europe. Of such cases, 5 to 10% are due to the inheritance of a gene that predisposes the individual to the development of the disease. After an intensive effort by several laboratories, two genes named BRCA1 and BRCA2 were identified in the mid-1990s as being responsible for the majority of the inherited cases of breast cancer. BRCA mutations also predispose a woman to the development of ovarian cancer, which has an especially high mortality rate. It was pointed out that cells possess checkpoints that halt progression of the cell cycle following DNA damage. The BRCA proteins are part of one or more large protein complexes that respond to DNA damage and activate DNA repair by means of homologous recombination. Cells with mutant BRCA protein accumulate chromosomal breaks and exhibit a highly aneuploid karyotype. In cells with a functional TP53 gene, failure to repair DNA damage lead to the activation of P53, which cause the cell to either arrest cell cycle progress or undergo apoptosis. We have seen in this chapter that apoptosis is one of the body's primary mechanisms of riddling itself of potential tumor cells. The mechanism of apoptosis was discussed in the last chapter, and we were pathways that promote cell survival rather than cell destruction. The best studied cell survival pathway involves the activation of a kinase called PKB or AKT by the phosphoinositide PIP3. PIP3 in turn is formed by the catalytic activity of the lipid kinase PI3K. Activation of PI3K to PKB pathway lead to an increased likelihood that a cell will survive a stimulus that normally would lead to its destruction. Whether a cell lives or dies following a particular event depends on a large degree on the balance between pro-apoptotic and anti-apoptotic signals. Mutations that affect this balance, such as those that contribute to the overexpression of PKB or PI3K, can shift this balance in favor of cell survival, which can provide a potential cancer cell with tremendous advantage. Another protein that can affect the balance between life and death of a cell is a lipid phosphate PTEN, which removes the phosphate group from the 3' position of PIP3, converting the molecule into PI45P2, which cannot activate PKB. Cells in which both copies of PTEN gene are inactivated tend to have an excessively high level of PI3, which leads to an overactive population of PK3 molecules. When a normal PTEN gene is introduced into tumor cells that lack a functional copy of the gene, the cells typically undergo apoptosis, as would be expected. Like the other tumor suppressor genes, mutations in PTEN cause a rare hereditary disease characterized by an increased risk of cancer, and such mutations are also found in a variety of sporadic cancers. Then DNA damage, BRCA1, BRCA2, failed repair, BRCA1, BRCA2, etc. are involved. So mutations or deletion is not the only mechanism by which tumor suppressor genes can be inactivated. Tumor suppressor genes such as BRCA1 or PTEN can often be rendered non-functional as the result of epigenetic mechanisms such as DNA methylation or histone modification, which silences transcription of the gene. Then oncogenes. As described above, oncogenes encode proteins that promote the loss of growth control and the conservation of a cell to a malignant state. So oncogenes encode proteins that promote the loss of growth control and the conversion of a cell to a malignant state. Then oncogenes are derived from proto-oncogenes, which are genes that encode proteins having a function in the normal cell. Then numerous oncogenes were initially identified as part of genomes of RNA tumor viruses, but many more have been identified because of their importance in tumorigenesis as determined in laboratory animals or human tumor samples. Different oncogenes become activated in different types of tumors, which reflects variations in the signaling pathways that operate in diverse cell types. The oncogene mutated most frequently in human tumors in RAS, which encode a GTP binding protein RAS that functions as an on off switch for a number of key signaling pathways controlling cell proliferation and metabolism. So, oncogenic RAS mutants typically include a protein whose GTPase activity cannot be stimulated, which leaves the molecule in an active GTP bound form, sending continuous proliferation signals along the pathway. Despite extensive efforts to develop anti-RAS strategies for cancer therapy, no drug that block RAS function have yet to be approved. The function of a number of oncogenes as summarized. So, oncogenes that encode growth factors or their receptors. The first connection between oncogenes and growth factors was made in 1983 when it was discovered that cancer-causing simian sarcoma virus containing an oncogene derived from the cellular gene for platelet-derived growth factor (PDGF), a protein present in human blood. Cultured cells that are transformed with this virus secrete large amounts of PDGF into the medium, which causes the cell to proliferate in an uncontrolled fashion. Overexpression of PDGF has been implicated in the development of brain tumors or gliomas. Another oncogenic virus, avian erythroblastoma virus, was found to carry an oncogene that encode an EGC receptor, that is missing part of the extracellular domain of the protein that binds the growth factor. One might expect that the altered receptor would be unable to signal a cell to divide, but just the reverse is true. This altered version of receptor stimulates the cell continuously, that is, regardless of whether or not the growth factor is present in the medium. This is the reason why cultured cells that carry the altered gene proliferate in an uncontrolled manner. A number of spontaneous human cancers have been found to contain cells with genetic alterations that affect growth factor receptors, including EGFR. Most commonly, the malignant cells contain a much larger number of receptors in their plasma membranes than do normal cells. The presence of excess receptors make the cell sensitive to much lower concentrations of the growth factor, and thus they are stimulated to divide under conditions that would not affect normal cells. As discussed below, growth factor receptors have become a favorite target for their therapeutic antibodies, which bind to the receptor's extracellular domain, and for small molecular inhibitors, which bind to the receptor's intracellular tyrosine kinase domain. Oncogenes that encode cytoplasmic protein kinase. Overactive protein kinase function as oncogenes by generating signals that lead to inappropriate cell proliferation or survival. RAF, for example, is a serine threonine protein kinase that heads the MAP kinase cascade, the primary growth controlling signaling pathway in cells. It is evident that RAF is well positioned to wreak havoc within a cell should its enzymatic activity become altered as a result of mutation. As with the growth factor receptors and RAS, mutations that turn RAF into an enzyme that remains in the own position are most likely to convert the proto-oncogene into an oncogene and contribute to the cell's loss of growth control. RAF is most closely linked to melanoma, where BRAF mutations play a causative role in the development of approximately 70% of these cancers. Another group of cytoplasmic kinase that are often deregulated in cancer is the cyclin-dependent kinase, especially CDK4 and CDK6. Cyclin D1, a regulator of the CDKs, is also a frequent oncogene. The first oncogene to be discovered, SRC, is also a protein kinase, but one that phosphorylates tyrosine residues on protein substrates rather than serine and threonine residues. Transformation of a cell by a SRC containing tumor virus is accompanied by the phosphorylation of a wide variety of proteins. Included among the apparent substrates of SRC are proteins involved in signal transduction, control of the cytoskeleton, and cell adhesion. For an unknown reason, SRC mutations appear only rarely among the repertoire of genetic changes in human tumor cells. Then oncogenes that encode transcription factors. A number of oncogenes encode proteins that act as transcription factors. The progression of cells through the cell cycle requires the timely activation or repression of a large variety of genes whose products contribute in various ways to cell growth and division. It is not surprising, therefore, that alterations in the proteins that control the progression of these genes could seriously disturb a cell's normal growth patterns. Probably the best studied oncogene whose product acts as a transcription factor is MYC. MYC regulates the expression of a huge number of proteins and non-coding RNAs like rRNA, tRNA, and miRNAs involved in cell growth and proliferation. When MYC expression is selectively blocked, the progression of the cells through G1 is blocked. The MYC gene is one of the proto-oncogene most commonly altered in human cancers, often being amplified within the genome or rearranged as a result of chromosome translocation. These chromosomal changes are thought to remove the MYC gene from its normal regulatory influences and increase its level of expression in the cell, producing an excess of MYC protein. One of the most common types of cancer among populations in Africa, called Burkitt's lymphoma, results from the translocation of an MYC gene to position adjustment to an antibody gene. The disease occurs primarily in persons who have also been infected with Epstein-Barr virus. This same virus causes only minor infections or mononucleosis in people living in Western countries and is not associated with tumorigenesis. Then oncogenes that encode proteins that affect the epigenetic state of cytochrome C. Two of the most important factors in determining the epigenetic state of chromatin are whether particular sites in the DNA or gene promoters are methylated or not, and particular modifications present in the tails of certain core histones within the nucleosomes of this same gene promoter. And DNA methylation tend to silence genes, whereas histone modifications may either activate or repress gene transcription. So recent studies have indicated that a number of oncogenes encode proteins that affect DNA methylation or histone modification. These include DNA methyltransferase, histone acetyltransferase, and deacylase histone methyltransferase and dimethylase, and promotes proteins present within chromatin remodeling complexes. Mutations in any of these classes of genes can promote tumorigenesis by increasing or decreasing transcription of genes involved in various signaling and regulatory pathways that affect cell proliferation, survival, migration, etc. To cite just one example, acute myeloid leukemia is characterized by recurrent mutations in DNMT3A, a gene whose product is involved in maintaining DNA methylation patterns during DNA replication. So a reduction to the level of DNA methylation could lead to increased movement of transposable elements, which would cause genetic instability as well as increased transcription in certain proto-oncogenes. So conversely, an increase in the level of DNA methylation of the promoter regions of tumor suppressor gene is known to silence the expression of genes that exert a key inhibitory influence on tumorigenesis. Next topic we will be learning tomorrow.